Thank you, Omar, for the introduction. And I'm really glad to present in this first BioArc workshop. As Omar mentioned, I'll present two works. One of them is purely when I was a postdoctoral researcher at ETH Zurich, and the other one is also a collaboration between AMD and ETH Zurich as well. So I'll first start with, I don't really need to give like a lot of introduction because Omar gave a really great talk on the overview of complete genome analysis, but still I'll get a bit of a refresher. As we know, until today, there's not a single machine that can read the entire content of genome. So we always have to break down the sequence into smaller chunks. So I would like to go with this slide, which is from Mohammed, one of our works, where we see a typical genome analysis pipeline. So we always start from a DNA sample, and which we generate from sequencing machine, and then there are different companies, for example, Illumina, ONT, Bio. These are the major companies. And then once we have the genomic data, then we start the genome processing step. And the very first step is basecalling, where we convert the raw sequencing data to ACGT DNA-based pairs. And for example, like this process over here, I show it from ONT, but similar approaches are used by different companies as well. Then we go through a complete pipeline of steps to perform read mapping to locate each read sequence in a reference genome. So where we perform indexing, seeding, pre-alignment filtering, and sequence alignment. And like Omar mentioned, traditionally there have been a lot of research on read mapping, especially the sequence alignment part. So we were also interested in focusing on the previous steps before sequence alignment because they can also become bottleneck. So in the very first work, we focused on basecalling. As I mentioned, it's the very first step in the genomic analysis where we use modern basecallers are using like complex deep learning based models. So for example, you have a sequencer, you give an input squiggle and then pass through like complex deep learning based models to, in the end, to map it to an ACGT base pair. And there is not really one-to-one mapping between the input signal and the output characteristics. So the overall sequence can only be decoded by aggregating over multiple time steps. So it's a bit complicated problem. So we were interested in accelerating and analyzing these deep learning-based basecallers. So in the very first step, we wanted to see like how these models perform if we prune a part of the model. So pruning is nothing but it's like a model compression technique where we discard network connections that are not really important to the performance. So if over here you see one of the state-of-the-art basecallers, which is Bonito, which is an open source basecaller from ONT. On the x-axis is the sparsity. Sparsity means like how much pruning we perform. So if we do 100% pruning, it means we remove complete weights of a neural network, and 0% is we don't really remove anything. And on the y-axis is the accuracy, and the higher the better. We perform two different ways of pruning. One is structured pruning, and the other one is unstructured pruning. Unstructured pruning is that we don't really keep the structure, and we just prune randomly from anywhere from different layers of neural network. This kind of pruning leads to really high sparsity, but with this pruning we know how much of the model is really not important, so which contributes very little to the model's inference or the accuracy. And structured pruning is more hardware friendly because we retain the structure so you can accelerate it quite well on any accelerator. And then we also have a figure which correlates to the model size over here. So when we perform pruning or unstructured pruning, how the model size changes. And we observe, like, even when we perform like 30% of pruning for this model, we see still the accuracy remains the same, which means like we can get rid of 30% of model using structured pruning. However, when we go for unstructured pruning, this becomes we can, without it, we lose loss in accuracy. And then if we want to further push the limits, like if you want to analyze how much more we can prune, we see with structured pruning, we get still the 60% of pruning, and still we.
Okay, where's John?
Recording in progress.
3% of the model we are performing basecalling, and still we are getting 85% accuracy. And if we see the model size, now we are left with only like 33.3 times lower model. This is because often these basecallers are adapted from speech recognition domains. Because the problem is kind of similar. We have like an input signal, which could be a speech, and then we need to map it to a vocabulary, which in our case is ACGT signals. And in speech recognition domain, the vocabulary is quite bigger and higher, so often we need like way bigger models. But over here, if you just take the model from speech recognition domain and apply it into both basecalling, it leads to often over-parameterized model, and we don't really need that. And when we go for hardware acceleration, an important effect is to analyze the quantization. So quantization is like reduction of the bit width precision and with the calculations are performed. So in neural network, we quantize both the activations and the weights. Over here, I have an example of one of the organisms. On the x-axis, we are seeing the quantization of weights, and we have the accuracy. And we have a similar plot for like model size, how the model size changes with different procedures. And we observe even when we go like 4 times lower, still we get almost the same accuracy. So there is not really loss on any accuracy. And in this case, we are using the same procedure for all the layers. But we could also have designs where we could have different procedures for different layers, and we can have a mixed procedure architecture. And even if we go lower, like we use 4 bits, still we hardly see any loss in accuracy. And even with the lowest one, which we evaluated, which is 3 bits for weights and 2 bits for activation, still we are able to base call with 82% of accuracy, which shows like we really need to quantize this model, and still we don't really lose a lot of information. However, most of the base models are using floating point precision to represent each of these neural network layers. So there's really a lot of room for improvement to accelerate this process. However, what do we fair there? Because there are so many considerations you need to think about, like different neural network modules, different layers, configuration of the layers. It could be different kernel sizes, what sort of resolution relation into the business one. The design space really explode. So that's why, in our work, our goal was to
we can open the door and see.
Recording in
In
progress.
this work, we introduced RUBICON. This is the first for special high.
It seems the transition to my network, but isn't it okay?
Isn't there a cage?
No.
I have a cable, but it's not that long.
I see. Yeah, I mean, let's see what comes up. I wonder.
Recording in progress.
Do you have dongle for the cable?
No.
I don't think they have either.
To evaluate millions of in neural architecture search, what we do is we always first define the search space. That's the size of
Recording in progress.
This is the search space. explode, but like we have billions of them. And then we add, during the QABAS, we need to provide the raw sequencing data, which comes from ONT, and they are like a lot of open source data sets. So that we go with the data sets and we update the model weights with the search process. And then we provide hardware constraints in terms of latency or in terms of throughput, so which, and you can also specify like what sort of latency would you like to achieve? Would you want to go for a rather inaccurate model, but it's able to satisfy a latency constraint, or you don't really care about the latency and you would like a highly accurate model? So you can provide this in the hardware constraint. And then during the search phase, we enable different edges and update the architectural weights. And so we go over different connections and we choose different modules. And for example, over here we have the first module, which is a 8x8 quantized multi-convolution, which is connected to another 8x4 convolution. And then in the end we have identity. Identity is nothing but it's just you just map the input to the output, so you can easily remove this block. So this kind of search space also helps us removing multiple blocks as well. So in the end, if you see, right, we are really not just trying to quantize the complete model into a single quantization space, but for each layer we are using a different quantization. So it really leads to a completely mixed procedure architecture. And now we'll go over a second block, which is SkipClip. As I mentioned, like now in deep learning based networks, we often have skip connection or residuals. For example, over here, which help to mitigate the vanishing gradient problems, and so they help to improve the accuracy. And so we see like a lot of basecallers have also started having a lot of skip connections. But if we think from the hardware point of view, it's not really good to have these skip connections because they increase the data lifetime. So if we have to store the data somewhere because we need to feed it later, so we'll have to buffer it, like we need like more storage, and also additionally these layers need to match the channel sizes, so you perform additional computation. And also it leads to irregularity because we have these connections which could span like multiple layers. It could be like 10 layers as well. So they're not really good from a hardware acceleration point of view. So that's why we wanted to get rid of these connections. So we provided another module which we call as SkipClip, which helps you remove skip connection which are present in the basecallers. And for this process we use knowledge distillation. So in knowledge distillation, what we do, we have like a pre-trained model, which we call as teacher network. So for example, you take your unproven, completely off-the-shelf model, and then we take a student network. For example, over here we have a model that comes out of QABAS. And then what we want to do is like we want to teach our student network to mimic how the teacher network behaves. So what we do, we start the SkipClip process. So in the first epoch, we perform forward pass for both of for the networks, and use the loss from the teacher network to update the weights of the student network. And this way we are just doing the backward pass for the student network. And then in the next epoch, we remove one of the skip connections from the student network and we train for the entire epoch. And then we do the similar way, we go for the next epoch and then we remove another skip connection. And we keep on doing this process until we get rid of all the blocks. And then we do several steps of fine tuning so that we are able to recover the lost inaccuracy. And we observe, if we do it this way, we are able to remove all the skip connection. So in the end, SkipClip gets the best of both worlds, that we are getting a highly accurate model, and the topology is quite regular. So we can really easily accelerate on any hardware accelerator. And these two blocks are like, we can use it together or separately, they're independent of each other. And then using these two blocks, we developed an example basecaller, which we call as RUBICALL, which was developed using QABAS and SkipClip techniques. If you see over here, RUBICALL has multiple blocks, which all of them are quantized, and we observed that the lower blocks, the earlier blocks use higher precision, which are using like 16 by 16 for both and weight and activation, while for the later blocks we only have 8 bits or 4 bits, which makes sense, because if you think about it, like from the And if you see from the input side, we have an analog signal which requires more precision, but on the output side, we just only need to map it to ACGT base pairs, so we can really do it with a lower precision. So QABAS is able to learn these kind of techniques. So it really performs hardware aware and also algorithmic aware search. And in the end, we have a CTC block which decodes the nucleotides to map it to ACGT base pair. So in the end, we have a really mixed precision architecture. And then we wanted to compare this architecture with other basecallers out there to see like how good or bad like our search algorithm was. So we compared with 5 state-of-the-art basecallers like from ONT and other open source basecallers like SACall, which uses a transformer, and Dorado Fast, which is quite state-of-the-art, where they're using LibTorch to use the tensor cores. But it's like the Bonito version of the CRF model, where they're using RNN. And then we evaluate 2 different versions of RUBICALL. One is RUBICALL MP, which is the mixed precision version, which I showed before. And then we also analyze the RUBICON FP, which is just using like traditionally like floating point precision, so you can easily use it on any GPU. And first we show the base calling throughput. So on the x-axis is different species, and on the y-axis is the base calling throughput, and we compare with different baselines. If we compare to Dorado, which is the state-of-the-art fastest basecaller, we observed that RUBICALL MP outperformed Dorado with 3.9x, which is because we are really using mixed precision, not just like a single specific precision for all of the cores. And then if we compare RUBICON MP with like RUBICON FP, which is using floating point precision, just by going to a mixed precision architecture, we are able to get 63x higher performance, and which is quite amazing compared to other basecallers which are still using floating point precision. And then we also perform hardware code design, and that really helps to accelerate the performance. And so if we compare with all of the basecallers, RUBICALL is consistently outperforming all the evaluated basecallers. And then we also wanted to see like the accuracy, because that's also an important metric. And over here we just have RUBICALL, instead of RUBICALL MP or FP, because they use the same model architecture, and in the end we get the same accuracy, so we just show RUBICALL. So RUBICALL provides 2.97 higher accuracy than Dorado Fast. So it's not just the accuracy, the speed, but also accuracy compared to the Dorado Fast version, which is the fastest basecaller. And then if you compare with the hand-tuned expert design model, which is the Bonito model, which is from ONT, we call it as expert design model because this model has the same backend as our model. We get way higher performance in both the versions, like in the floating point version and also the MP version, which is the mixed precision version. And we get similar accuracy for like similar gains compared to other baseline approaches. So if we want to compare the overall results, so we want to plot it in terms of different metrics to get a Pareto plot. So we have base calling throughput on the y-axis, and on the x-axis the parameters. How many parameters are there? So parameters kind of correlate with how much calculations or multiplications or convolutions you need to perform. And then we do it like similar for the model size. So the lower the better on the x-axis. And then we have another metric, which is the base calling accuracy. Not only the base calling accuracy in the reverse order. So if you see the Pareto point always lies on the left side, the higher the better. And if you see for the parameters, Dorado Fast has the lowest amount of parameters, but still it gives lower accuracy, lower throughput. This is because RUBICALL MP is using like really lower precision than what Dorado can do. It's providing way higher performance. And we see a similar trend for the model size. However, if we compare to the accuracy, as I mentioned before, we are getting 2.9% higher accuracy with using RUBICALL. So RUBICALL provides the ability to base call accurately, quickly, and efficiently. So it can easily lead to reduction in model size and neural network model parameters. So in our second work, I'll quickly go over the pre-alignment filtering work, which Omar mentioned. This work was which was published in IEEE Micro. As Omar mentioned, like we spent a lot of time, execution time on read mapping, which is mostly on dynamic programming step. However, overwhelming amount of these candidate locations are dissimilar, so we have to discard it. So it doesn't really make sense to perform this dynamic programming step. So if we can get rid of these dissimilar candidates before, it would really help accelerate the read mapping step. So therefore, we have these pre-alignment filters that do exactly the same. And the Sneaky Snake is state-of-the-art pre-alignment filter, which is from our group from Mohammed. And the key idea, I will give a really high idea about like how this accelerates pre-alignment filtering is by approximating the edit distance calculation, which is similar to a single net routing problem in VLSI. So, for example, we have a VLSI chip over here, and when we dissect it, then we can find some components and link around the chip. And the problem becomes similar to like the snake traversal through a maze. So the snake wants to go out of the chip layout or the maze, and we need to find the shortest path, how the snake can progress. And there are like more details about probably which Mohammed will discuss about. And in this work, our goal was to analyze the performance of this Sneaky Snake on a server-grade system and see how we can accelerate this algorithm. So over here we have a roofline plot. On the x-axis is the arithmetic intensity, and on the y-axis is the performance. If we plot the Sneaky Snake on this POWER9 system, we observe that this algorithm is heavily memory bound and it has complex memory access patterns. Therefore, it leads to limited performance and high energy consumption on CPU-based systems. And so therefore, our goal in this work was to mitigate the performance bottleneck of these modern pre-alignment filtering in an energy efficient way. And we also wanted to evaluate near memory acceleration on real hardware using state-of-the-art interfaces like OpenCAPI. So in this work, we built the first near memory pre-alignment filtering accelerator using an actual system which was connected to which we have a POWER9 system with an HBM-based FPGA board. And we optimized this accelerator using data-centric caching scheme where we have heterogeneous memory hierarchy using URAM, BRAM, HBM. And we evaluate different systems as well, not just HBM, but we also evaluate the traditional DDR4-based system. We evaluate different interconnect technologies like CAPI2, OpenCAPI. And we also try like scaling of processing elements using a single channel or multiple channels, because the HBM-based boards, they provide a lot of channels, like up to 32 channels, which we can exploit. So we wanted to study all these different characteristics of the board. So in our contribution, we also include the scalability analysis. So if you see over here, we have the performance results for runtime for a different number of processing elements on the x-axis. And we have different configurations using HBM, using OpenCAPI interface, or if you're using multiple channels or single channel, or using different, or we are using DDR4 technology. And we also do a comparison for energy efficiency similarly. And then we observe that near memory acceleration improves performance and energy efficiency by up to 27x and 133x respectively over the complete CPU-based system. And we also provide a lot of insights and observations. For example, this HBM designs, they can avoid the memory access congestion, which is typical in DDR4-based FPGA design if we scale properly with the number of channels available. And then we open source our design, and please feel free to try out on our GitHub page. So there are many more details, and I recommend you to read our IEEE micro paper for that. Before I conclude, I would like to come back to this nice picture. As I mentioned in my talk, we accelerate 2 steps in the pipeline and demonstrate like great results. However, like Onur was mentioning before, the task is still not over. There's still a long way to go, because the ultimate goal would be to perform end-to-end genome sequencing pipeline on a single device rather than using different state-of-the-art approaches where we perform different steps. And we are seeing in industry and in academia a progress towards that direction. At the end, I would like to acknowledge the help and support from everyone. Thank you.
Okay, thanks, Gagan. Any questions to Gagan?
Sorry, I went a bit over time.
Yeah, but we had some connection issues, so we couldn't catch you everywhere. Hopefully it was better on YouTube.
Okay.
I think it was better on YouTube.
Okay.
Pardon. Okay. I assume there are no questions. Do you have any questions?
we can have one quick question.
Okay.
Maybe I can ask.
Go ahead. Yeah.
Hey Gagan. So I have one quick question. You were showing these RUBICON FP and I guess MP results.
Right.
And FP looked significantly slower than the other one, and you said their accuracy is same. So could you quickly describe what are the potential use cases of FP if the accuracy is same but it's slower?
Right, it's a good question. First I'll mention like why the accuracy is same, because in our goal was to keep like throughout this when we were trying to develop a example basecaller, our goal was to keep the accuracy same. It doesn't matter if we go for lower precision or we prune the model, and that's what we observed from our like the motivation slides which I showed, that we can really push the accuracy, push the quantization and still keep the accuracy. And that's why with these two models we always get the same accuracy. And the potential use case for this FP is that you can really use it out of the box on any GPU. So because like we are using a lot of these basecallers on GPU right now, so that's why we build like one FP version, so you can run on GPU. While for the MP, it's like mixed precision. So right now, we don't really have the support on the GPU to exploit from, at least from the high-level library, like from PyTorch or TensorFlow. You can't really accelerate like a mixed precision, dynamically adaptive, low precision cores. So that's why and I mean, if you use like a libtorch, which is like a C++ interface to the tensor cores, then you can exploit these low precision units, but at the moment, it's not possible. That's why we wanted to have two different versions, so that one is like kind of a bit more specialized, so you see what you can achieve, and the other one is more like what you can use out of the box, and it kind of shows like what you're missing on, like amount of performance which you're losing just by staying on floating point. And if we see like ONT has already started moving towards using libtorch so that they can exploit these low precision units, so we see that even these companies are realizing that we need to kind of move towards squeezing every inch of performance from it.
Thanks a lot for that.
Thanks, John.
Okay, thanks, Gagan. I think we should move to the next talk since we don't have much time. Next up is Leonid. Leonid, can you hear me?
Yes, I can hear you.
Okay, great. We can hear you and see you also.
Very good.
Okay, let me introduce you quickly. So Leonid is with Bar-Ilan University. Before that, he was at Technion. He's been doing work on genomic acceleration using especially new technologies like RUBICON in the past. And he's going to talk about some of the work that he's been doing on hardware-accelerated detection and classification of pathogens of epidemic significance. So Leonid, please go ahead.
Right, thank you very much. I will try to be quick. Good morning, everybody. And Ramadan Kareem to our Muslim participants. Let me give you a quick introduction to our lab. It's called ENIX with the engineering department of Bar-Ilan University. We have 6 faculty members and 100 researchers and students, including grads and undergrads. Many research topics which I'm not going to cover. You can take a look. I mean, it's easier to find things which we're not interested in. An interesting fact about ENIX, 50 plus tape outs in 8 years of its existence. Some of those chips are large system on chips with application processors and several heterogeneous components. So that's an interesting operation and something worth looking at. First, let me start with the motivation, and I have a very long motivation which is divided in several parts. The first one is viral pandemics. like the one that we have completed recently, or maybe we just learn to live with that because it's still lurking out there. We just kind of pretend it's not there, but it is there. But let's imagine that in a few short years, the world is struck again by a new viral pandemic. Many people agree that it's going to happen. We don't know when exactly, but there's very little doubt it's going to happen again. And let's assume that in this new pandemic, the virus is going to mutate as quickly as it did in COVID-19 pandemic, creating many hundreds, maybe thousands of viral variants and lineages. One of the lessons of COVID-19 is that polymerase chain reaction test, which is the main diagnostic tools used today, is not really adequate for serious variant discovery, because first, it doesn't know, it cannot discover new variants, and second, to classify newly obtained samples into existing lineages or families, it requires so-called primers to be designed per lineage, and that's an expensive and very lengthy process. So assuming that PCR can be used for classifying a viral genome into hundreds, maybe thousands of existing lineages, that's kind of impractical. So the ultimate solution for that is DNA sequencing and then computational analysis. Now the problem here is that this is a very large-scale task. Again, based on the lessons from COVID-19 pandemics, we can easily assume that we're going to test hundreds of millions of people daily during the next viral pandemic, of which 10–15% may test positive, of which we're going to again sequence 10–15% to create statistically significant results. So we're talking about hundreds of thousands, potentially millions, maybe even tens of millions of viral genomes sequenced daily. Again, it's not something which is completely unreasonable given the progress of sequencing technologies, and we would have to process all those newly sequenced genomes in as wide context as possible, because it's not going to work out if we only will compare a newly sequenced genome with one single reference. We know that now. So that's, again, a problem very large scale. Another motivational component is called sepsis, which is a quick blood infection that typically happens, for example, in a hospital environment where people undergo surgeries and all kinds of procedures when their immune systems is weakened. So it's completely plausible that a person can undergo a complex operation and come out just fine, and then develop a blood infection, which can cause severe illness or death. I was amazed myself when I learned that up to half of hospital deaths in the United States alone are caused by sepsis. Now, in sepsis, early identification of the microbial pathogen that is responsible for this infection is absolutely critical. If we cannot find out quickly the nature of the pathogen, quickly meaning in hours, maybe a day, then it just might be too late. And what happens in real life, not only we cannot identify them quickly, in a third of the cases, the underlying pathogen is not even identified. So the result is that antibiotic treatment is not pathogen targeted. It's rather more epidemiological, meaning you just decide on treatment based on what goes around these days. And all these clearly leads to antimicrobial abuse and overuse, which again leads to our third motivational component, which is closely linked to the second one, and that's called AMR, antimicrobial resistance. And that's an overly, continuously escalating global crisis, which might not be as well known as a viral pandemic, but it's probably as dangerous, maybe even more dangerous. World Health Organization, for example, defines AMR as one of the top global public health threats that are facing humanity. AMR causes at least 700,000 deaths a year and is expected to grow extremely significantly in the next 25-30 years. Again, AMR is caused by our inability, it's not caused by our inability, but one of the reasons for AMR is our inability to understand quickly, to identify quickly microbial pathogens that cause all kinds of infections and inflammations, and as a result, the treatment again is more epidemiological than pathogen related, and then antimicrobials are overused and abused, which lead to more AMR. So to summarize the pain, accurate pathogen detection classification is a time-critical problem, both in viral pandemics, when we talk about a viral pathogen, and in antimicrobial resistance in all kinds of blood infections, in all kinds of bacterial infections in general. Again, time-critical. It's a problem of very large scale, especially in viral pandemic, where we have tens and hundreds of millions of test samples daily, but also in bacterial pathogen, because it happens everywhere in the world, in every hospital, in every clinic, to every patient. Traditional methods, which range from PCR and petri dishes and growing samples and growing cultures, through the computational methods that are in use today, they are either prohibitively slow, even on high-performance computer infrastructure, or they trade accuracy for speed, which make them less accurate. So that's the pain that we would like to resolve. Our solution is a real-time hardware-accelerated pathogen classifier. It's hardware-accelerated because, as we know, as Honor explained to us very eloquently, it's unhealthy to have a special purpose machine to generate data and to have a general purpose machine to crunch it. So to work as fast as the sequencer does, we need a hardware accelerated solution. The input to this solution, to this platform, would be a set or a stream of unaligned DNA bits which are sourced from a metagenomic sample. Well, that's the real-life scenario when we have a sample which contains mainly the host DNA, but also lots of DNAs of all other organisms that can be found in blood or spat or a urine sample. And some of those organisms can be, pre-processed, some of those DNAs can be amplified, for example, some maybe not. So we need to deal with metagenomic samples, and we preferably don't want to align the reads and assemble the genome to apply the classification. And yet the output we want to first detect the pathogen, the target pathogen that we're looking for, or several target pathogens. We want to classify the pathogens that are found in a sample into existing classes, or to signal that a new variant, for example, of a viral pathogen is discovered. Or we want to profile the pathogens. If there are several bacterial and viral pathogens in a sample, we want to, build a profile of pathogens that we find. The requirements for such a platform would be real-time operation. So if we're talking about nanopore based sequencer, that would be the speed of sequencer, and if it's a Illumina class sequencer, then as fast as possible. And we would obviously require high accuracy and all those criteria that comes with accuracy, sensitivity, precision, specificity, all those. And we would envision this classifier as a portable device. It's not the only embodiment, obviously, but the preferable embodiment is a portable device which works in conjunction with a portable sequencer and provides the answer quickly and accurately in a field environment, for example, in small clinics, in labs, everywhere where samples are tested. That's the idea. Two different solutions which we have come up with, and I can discuss here. So it's a little busy slide, so let's look at the right top angle first. So this solution is based on a special kind of memory which is called an associative or content addressable memory. And to some of you, maybe most of you, who don't know what that is, so in addition to read and write, content addressable memory, unlike normal random addressable memory, can also search or look up or compare a search pattern with the entire content of the memory in a single clock cycle. So it allows you a very, very quick search. Associative access is used everywhere in computer architecture. Everybody knows what cache is. So cache, for example, is a kind of associative memory, maybe not fully associative, but it's a kind of associative memory where we perform a search operation, we search for a tag in every cycle. So this solution that I am talking about here is called Hamming distance tolerant content addressable memory, or HDCOM, and a pathogen identifier which is based on this HDCOM. So here at the center, that's the illustration of a classification pipeline based on HDCOM. So we have a sample, and we have a sequencer, and we have a bunch of unaligned DNA reads, and we feed them into HDCOM, which prior to that, we built a reference database, and the reference database, again, built offline, placed in the HDCOM, which is a memory, and the reference database can be built linearly or as a graph, and it can contain several species of bacterial and viral pathogen. Can be built using k-mers, for example, or other DNA fragments. We don't need to go into that right now. This is more of a circuit work, rather than algorithmic work. So most of the innovation here is in the circuit level. This design was actually taped out, so we have a silicon prototype for this design. And at the bottom, you can see that's the chip. It's actually a multi-project chip of the kind that I was talking about earlier that we tape out in Enix. So it has, I think, like 12 different student projects. One of those is HDCOM, and this is a evaluation setup. So that's something which exists in silicon, and we are developing a bunch of different applications which have to do with genome classification, both in viral and bacterial directions on this silicon prototype. Another solution that I would like to briefly cover is called EDAM, which is an edit distance tolerant pathogen classifier. And unlike HDCOM, which tolerates Hamming distance, which allows it to tolerate sequencing errors and small variations, EDAM is utilizing the observation that most most errors and variations in DNA are actually edits, not simply replacements. And what you can see here in the middle part, it's an illustration of how a single edit can create a huge Hamming distance. So if we want to tolerate edits, tolerating Hamming distance is not enough. We need to build a special hardware which accounts for edit distance, not just Hamming distance. And that's what we did here. Again, I'm not going in deep into design. So here in the center we have a similar looking pathogen pipeline, but the solution over here is based on edit distance tolerant memory structure rather than Hamming distance tolerant. Here at the bottom right, you can see the base cell, which is kind of complex, very large, so it's not easy to scale, for which we have a different solution, which is based on resistive memory that allows much more dense implementation. And at the bottom and the center, you have a quick comparison of EDAM performance, meaning actually accuracy, precision, sensitivity, and F1 score compared to Kraken, which arguably one of the most popular classifiers today. It's a software tool, and what you see here is sensitivity, again, precision, and F1 score versus EDAM, edit distance threshold, the ability to tolerate edits. And if you look at F1 score, for example, there's a clear optimum point at which we strongly outperform Kraken, again, because Kraken uses exact matching versus database, and we allow edit distance, which contributes to both sensitivity and precision, and ultimately leads to a higher F1 score. That's about it. Just one last message here. We have a sister workshop, which is called AACBB, Accelerator Architecture in Computational Biology and Bioinformatics, which happens in conjunction with ISCA conference in June, and that's actually the same day as honors PIM tutorials, so please consider contributing and participating in this workshop. Thank you.
Okay, thank you, Leonid, for the nice talk. It would be better to see you in person here, but I know you're teaching, etc. Yeah.
Yeah, sorry about that.
That's okay. Any questions to Leonid? Actually, I have a question. Why RRAM? For the last work you mentioned.
Why?
Well, yeah, why resistive RAM?
Only because of density.
Okay, I see.
Yeah, for example, if you look at EDAM and you have to go and look left and right to make sure you account for edits, in ReRAM you can simply create 3 different copies of the same fragment. One is like centerpiece, one shifted left and one shifted right, and because it's so dense, you can do it in a single cell, so you not only getting a denser solution, you also lose all these finding connections left and right, which create wire-bound situations. So that's the reason.
Okay, it makes sense. Basically, you get a lot more density. Okay, thanks. Any other questions?
My pleasure.
From the audience? Yeah, I will reiterate that if you want to go to ISCA, Leonid's workshop is definitely worth submitting to, and I've participated in that workshop many times. I intend to be present in 2 workshops this time, concurrently.
Yeah.
Okay.
We're looking to see you in both.
Yes. Okay. Thanks a lot, Leonid, again.